Good afternoon, everyone. Good evening, depending on where you are joining this webinar from. A very warm welcome to everyone who joined the webinar today. Um, my name is Subrata Bhattacharya. I'm the founding director of PharmaQuant. I am a pleasure to host this webinar for everyone in the team. I would like to introduce my colleagues who are joining me today with. I have Ian Fotheringham, who is joining not very far from where I am, from UK. And Ian is a very seasoned expert in evidence synthesis and value analysis. So he will be doing the bulk of the talking today and walking everyone through ActiveSLR. I'm also being joined by my colleagues in India, Sohel and Anup. Sohel has been fundamental behind design of ActiveSLR, and Anup took care of all the IT requirement for ActiveSLR. So thank you to both of them for their continuous effort on this. Without Before I go to hand this over to my colleague Jan, I just wanted to give a little context on why ActiveSLR. So one of the reasons we created ActiveSLR is because we saw the need for a product like this. Now there are several other products available in the market, which is very equivalent to what ActiveSLR can or cannot do, but all those products are um, behind a paywall. So you need to pay on either monthly or annual basis to get access to a product like ActiveSLR. Um, Now they are also probably as good as ActiveSLR, or other way put, ActiveSLR is also as good as those products. But because of the paywall, many of the researchers across the world, primarily from low and middle income countries, cannot have access to a useful tool like ActiveSLR, which will expedite their research work. So we were using this ActiveSLR tool for quite some time internally. So we thought that we will make this tool available for free use to the rest of the world so that people can take advantage of all the advancements that we have made on the information technology side. So this is completely free to use. So please use it as much as you want, and please let your colleagues or let your other team members also know that this is a free tool to use, and you can use as freely as you want. Without further ado, I will hand over to Ian, my colleague. Over to you, Ian.
Thanks so much, Rata. And yes, very good morning, afternoon, or evening, all. Very good to have you here. So the plan today was really just to give you a quick rundown of the app. Appreciate that this is brand new for everybody. Thank you to all of those who have signed up and are starting to use it already. But this is our first webinar just to show you how to use it, how to get set up, and some of the functions that we have on offer at the minute. So I'll start sharing my screen. And when you first join, head over to activeslr.io, our website. This is the sign up, the homepage that you'll see. You have a little bit of information here about what it offers, and also there are some details on the sign up options. As you rightly said, Shibata, it is entirely free for wherever you're joining from. If you have a Scholar account, that's really for the academics, the universities among us. Corporate is anybody, a consultancy, a pharma company, biotech, anybody else. Everyone is welcome to use this. The only real difference between the two is that we have identity verification, just like an email check to confirm that you're signing up from an appropriate email address, so that nobody's imitating a company or university. So once you get signed up, you will be able to log in, which we'll do now. Now, the first thing you'll be able to do is to have this page that will show any teams that you have joined. Now, one thing to note is that you are able to join different teams, different larger groups of projects. This could be helpful, for example, if you're using a vendor, if you're partnering with another company, maybe you want to jump into one of their team that will allow you to access one of their projects if they're kind of running it. So there's an opportunity there to have multiple setups, multiple teams that will only show the projects that you are part of, but that you may well be kind of in a slightly different setup. So we're just doing a demo set of projects today. And so I'll go into the demo corporate account that we've set up. Now, when you log on, this will be something like your dashboard. Obviously, you won't have any project set up, so this will be blank. If we click on the ActiveSLR logo, you have more information, which will be handy in about a minute or two. Now, what you want to do first is go to your settings. We want to set up ActiveSLR for how you need it. And this is how we do it. So firstly, we can invite any and all users that you need for your projects. So you just simply add a user, pop their email address, and that will send them an invite that will go to them. They have to accept it, but this will then allow you to manage your users as you wish. So we've got a few people on this example project already. You'll note as well that we've got some different notations here about admin and user. Now that'll be where we go to next. So what we have done is set up a series of roles and responsibilities and divided the different access levels for ActiveSLR according to those roles and responsibilities. Now, I wanted to give you an example to show this first. So we have like project level permissions and also app access, software access permissions. Who can invite and delete users, for example? Who can, add projects, those types of things. Within the project permissions, we have 3 normal presets, but you can create as many as you wish of exactly how you wish. So I just wanted to first explain that when you sign up, you won't have this demo super user. This is one I just set up, but you'll have manager, reconciler, and screener. Now, within PharmaQuant, these are the kind of levels of permissions that we have. We normally have, for example, a screener that doesn't need to upload the papers, that doesn't need to see the dispute resolution, the quality checking. Maybe they're just a screener. They're not even doing data extraction sometimes. So the level of access that a screener needs is different to a manager. But you can set these up however you wish in the roles subheading. Just simply add a role. We can put test, and then this test will automatically pop up in your permissions tab, and you can create these however you wish. There's no limit on these. You can make these however you wish. You can have different permissions for different projects, different clients, however you want to do it. You can edit and delete as you need, and then they disappear straight away. So we have this set up to begin with. You then want to create a project. This will be blank for you again because you don't have any projects yet, but just simply add a project. And here we can put down a summary of the name, description, and a few of the key points like, do you want this single or double blind screening? And do you want this single or double blind data extraction as well? If you're doing single screening, we also have a QC threshold. This will become something that you can do 0 to 100. At the minute, we've set it at 20 to 100, but we will change that soon. But this is just that threshold, the minimum number, the minimum percentage of abstracts that you need to QC before you can accept it. That will be totally optional though. The other useful thing you can do is select the type of QC that you want. So if you're a project manager who's reviewing people's decisions, you can make it totally random. But equally, if you know you don't have many includes, let's say, you may want to move that to stratified so that stratification will ensure you get a proportion of includes and excludes that is relative to the decisions that have been made. So if you got a 20%, you will be guaranteed to pick up at least 20% of the includes. It will also stratify and sample the exclusion decisions. So if you need to make sure that people are excluding according to certain reasons, For example, some pharmaceutical companies will review the exclusion list for that very reason. You may want to do a stratified QC just to check that people are excluding according to patient population correctly, for example. So once you have a project up and running, you'll have a dashboard that will now show a project. I'll move over to this one just for a second. We've got two examples to show you. But as you can see on the left-hand side, this is your menu that has now appeared. Because I've set myself up as a super user who has access to everything, I can see the full list of tasks and functions that are available within ActiveSLR. Now, what we hope is that this reflects the chronology of the... Oh, sorry, Shabata, you're about to say something.
I just very quickly wanted to say that we have Q&A open. So while Ian is presenting, if anybody has any questions, please do not hesitate to post that on the Q&A section of the webinar today. And then once Ian is done with the presentation, we'll head to the Q&A section. So just wanted to say that out loud.
Very good point. Totally forgot all our preamble. Absolutely right. Thanks, Shabata.
Over to you.
Thank you. Yes, perfect. So yeah, we'll try and keep this to about 30 minutes. We've got good time to discuss afterwards. But anyway, you got your list of tasks here. They should follow the chronology of your literature review. Though at the end you do have a report section that gives you some PM metrics and also your final inclusion list. So we get first to our upload files stage. Here you can drag and drop, or you can just select this and choose whatever RIS files you want. That's the standard output for Ovid, Embase, PsycInfo, whatever you want. We don't know of any databases that are regularly used that don't allow a RIS output format. But if you do have one, please let us know, and we can look to try and accommodate. But here you want to upload your files from the search. Search, this is really where ActiveSLR starts. Once you have your searches, they can be imported into here. So what we do first is we look to make sure, cross-checking that the import count is correct based on the exports that you expected from your searches, and then run deduplication. Now, I have already done that for this project, but what you'll see is that this is the first AI assistance tool that we have in ActiveSLR. Now, you have true duplicates and possible duplicates. True duplicates are a far more direct cross-check of whether all of the words are identical. There may be some very, very minor changes. So, for example, you can see that Journal of Clinical Oncology in this example is not capitalized. So the algorithm still does identify that as a true duplicate. These are true duplicates. It even recognizes that some of the syntax that's coming through from this Ovid output is erroneous from the output from your RIS file that's wrong. So it recognizes that and allows you to identify these true duplicates. Now, what we expect as well is that what we have found is that if you do deduplication, for example, in EndNote, it uses a far more simple approach to identifying duplicates. It'll identify fewer, but we've also found in the past that sometimes it will erroneously identify a paper as a duplicate when there isn't a duplicate there, thereby accidentally excluding that paper. So you can have type 1 and type 2 errors in deduplication. So the aim here is to make this safer and better. So what you can do here in here, as you'll see, is you now have a list of 282 potential true duplicates that we expect you should find they are all duplicates. We've also got these possible ones. That's where the large language model, the AI assistance, has determined that there is a reasonably high likelihood of these being duplicates. You can see based on the title, it looks very, very likely, but this is where you can go as a user, make sure you scan through each of these, select which ones to discard, and hopefully this will speed up this will help speed up screening, because then you don't have these papers built into your actual screening later down the line. We've got a color coding system you can see here that will make sure that it helps kind of spot which ones are actually going. You can always keep all as well if you need. So that will then ultimately get you to your screening list. Now, one thing to note is that we will have our studies list here. You can export at any point. This will immediately export it to Excel. So at any point throughout the process, you have that kind of the ejection button at the ready. Hopefully we don't need it, and we'll move into screening. So this is the layout of our screening page. Now we intend it. The intention here is to try and keep it as clean and as focused on the material that you need as a reviewer to make that decision. One thing we have found is that some of the offerings available out there are really cluttered. And as a reviewer, as somebody who's had to sit through and sift through thousands of titles and abstracts, from my personal perspective, I find that you need to have a very clear understanding of the abstract and the title with little interference of everything that's out there. So this is what you have here, very intentionally so. Now, you will have set up as part of your project setup, you will have set up a little bit of a small PCOS. This is something that you can see here. Now, I'm just going to move over to other example. This is a psoriasis example that we've been using. Anybody who was in ISPO Montreal might recognize this. We had some really good demos on this. But what you'll see is that when you create this, the project, you can put in a review question and your PCOS. This I button, the information button here on the right-hand side, will be there throughout the projects, and this will be helpful for the user. Just pop it in. The exclusion reasons here are fully customizable as well in your project. If you wanted to at any point, you can go and set this up as part of your project. In the interest of time, I won't do that on this one, but we will set up a webinar. We will set up a series of how-to videos on YouTube, which can show you that. So we will have a look at the screening. You can exclude. We offer presets of PCOS, population intervention outcome, intervention comparator outcome and study design. Also sometimes have a language one as well. Now, within here, there are a few different user aids that we can offer. Now, the first one is highlighting keywords. Now, you as a user can choose your keywords that you want to have auto-highlighted on all of your references. This is purely bespoke to the project. So this is a psoriasis example. So we might want to look at PASI. It's a psoriasis activity, severity index score. You can choose whatever words you would like. You could have safety, and you could put that as green, whatever you'd like. This is fully customizable. If you run out of colors, you've used too many keywords, frankly, you can have hundreds of thousands of colors. And then that will automatically pop up in this and every subsequent paper. The other thing we've got is AI Pico. Now, this is links to your PICOS table that we have over here that was set up in the project. When you select this, this will start to auto-highlight materials based on the PICOS table that you have put together. So if I were to change this, if I was to specify that the intervention must be infliximab, it would not highlight the same level of interventions that you would see here. Now, the other cool thing here is that this is where another alarm comes into play, where your stringency can change. Now, in this example where we have it looks like some type of fairly robust clinical trial that is very kind of closely aligned to our PICOS. Maybe we want to increase that stringency, so you get rid of some of the noise there. This will increasing the stringency will hopefully restrict the amount of highlighting to only the most relevant information by raising the bar on the AI assistance for when it should highlight something for you. Conversely, if you found a paper that had very little, maybe it was like a more complex screening, you might want to just reduce that stringency, and the LLM will use a more inclusive approach to highlighting according to your PICO. This can change from project to project, paper to paper, person to person. That's however you wish. The other thing to note on here on our screening, this is how I think 95% of us do screening. You look at it paper by paper. If you wanted to, you could always do like a bulk search element. You could look for ustekinumab's, it's one of the psoriasis drugs. You can identify how many papers have mentioned ustekinumab, and you could do some type of like bulk decision on them if you knew maybe that was an intervention that you weren't interested in. That option is there if you wish. You can also revisit any and all of your decisions on this My Screened Studies subtab as well. So next thing then, once we've made some decisions, would then be to QC it. Obviously, this would normally be different people, but just for the purpose of this demo, as a super user, I can also perform the QCs. That's very much one of the reasons why we would normally have different people on different roles, but whatever you need as a user, hopefully you have the flexibility for it. So this was a paper that was obviously excluded for study design, given it's a narrative review. I think we would probably accept that. You have an option as well to accept all. So once you've gone past your 20% threshold, you'd be able to just press accept all and say, right, okay, let's move all of these on. I'm comfortable as a project manager, as a QCer, that the reviewing team have done their job. Now, at this point, one thing you might ask is say, okay, right, how do I know how far through that percentage of checks we are? So I might just jump momentarily to our project management tool, which is in reports. Now, because this is just an example project, the metrics that you see are not particularly inspiring, but if you had 3 or 4 people screening and were doing so at a reasonable pace, this would be helpful information to see how people are doing, how quickly are they working, how are people faring compared to one another. You also have decision metrics further down, and also this larger table that you can separate things out according to the reviewer versus the QCer to see whether there are major differences. And we have tabs for title and abstract, full text, and data extraction as well. In this title and abstract screen, this is where you can see you have the percentage threshold, and you can see how far you've progressed with those decisions. So this tool is there as PMs needed. So once we've done our quality checking, we can then hopefully move to full text screening. Now this is where another part of our AI assistance really comes into play. You'll see that we have icons on the PDFs. Now, one thing you will do is if you press find full text, that was very quick. It normally takes like 10 seconds or so. But what we're doing is as soon as you press full text, ActiveSLR is searching online for PDFs that are available for available to you as users. So the aim here is to reduce your time of having to go and manually hunt down freely available full text from PubMed or wherever you can get them. Now, we've got a few different icons that you can find here. So you can see if you have the PDF, then it's going to be available. It's already uploaded to you in ActiveSLR. You're ready to full text screen with the PDF there. If you have this pay button, this is where it is behind a paywall. And if you were to click, this is an example we set up, but if you were to click on the title, automatically takes you to a Google search. You can find it, and this is where you would go and pay for it. You could then, at any point, if you had a paper that you didn't have available, you could choose a file. You could upload a file, and it would automatically pop up here. Or you could flag this as a paid article if you knew that this one wasn't available and you needed to buy it. So this should be like a very helpful management system to help very quickly identify a freely available full text, but also manage that process, which can get a bit messy of looking for things available and then uploading subsequently. So once you've done that, then you can start working on your full text review. Actually, that's probably a bad example. I'll use this one that does have a PDF. So here we can now cross-check. We can now perform our full text review. The aim is very much to do this as we did before. You have your abstract here that you can apply that AI Pico to how we did before. but you also have the PDF now here as well. There are a couple of helpful tools here as well. Now, firstly, if you were doing this scoping, you realize this is a very relevant paper. Let's just move the decision and stuff out of the way for a second, and let's really focus on this paper. If we wanted to, this is another AI tool that will allow it that a large language model will draft up a PCOS for you from this full text. It will read it and it will draft this. So this is helpful if if you're doing some scoping work, you wanted to scan through some hits, you find a very relevant full paper, cross-check this auto-generated PICO with the PICOs that you're preparing to use. The other useful thing you can do is you can highlight in the full text. This will highlight all of the highlighted keywords that you have added previously. So this can be very helpful as you're doing a full text review. But the aim is really just to make that decision as we had done before. Once we're doing a full text review, look at the PDF, make your decision, and exclude or include accordingly. Then you have the exact same quality check as we did before. This is one that we included that didn't have a PDF, so maybe we want to exclude that by study design and make that correction. And then finally, we'll get to our included list. As before, you can export all of these to Excel. That's available to you at all times. And then as well, if anyone would like it, we do have data extraction in here as well. In this data extraction table, we have a form setup which can be populated as you need. We're basing this mainly on those kind of summary tables that you would get in a report, but the export functionality goes straight into Excel like we would normally do with a large Excel-based DET. We start you off with some standard treatment characteristics, but these are all fully customizable exactly how you wish. We were doing some example data extractions, so when you're working on it, you freeze the pane so nobody can make the edits. But if you need to add something as you go, you can do so. You can just simply add as you wish here. When we're working on these on the data extraction table, you can also make any limits you want to the data inputs. It can be yes, no, you can have a percentage, you could specify that, decimal points if needed, or, date and time, those kinds of things. So there's full functionality there. And once you're ready, you can freeze your form and then get data extracted. Now in here, we can, as a QC, as a super user, I can see what's in progress, what is pending. These things can start to be extracted. And if I, as a user, want to get extracting, I can just check this out, almost like a library system where you can check out this paper, say, I'm going to go and extract it. Then nobody else will be able to do that as you are data extracting. Once it's completed, it will then go into this tab, and as a QC, I'd be able to do some quality checking if I wanted to. I could look through our decisions. You can open the PDF if you want to make sure that everything's available to me here. I can cross-check this. It is indeed PAP 2025. That's a good start. And we can make our decisions as we want here. So, that's a really quick summary. I wanted to make sure that we had sort of 20 minutes or so to go through any questions anyone had. The only thing I wanted to offer just before we go is that you do have the option to set this up as a double-blind data extraction. You'll see here on this example that we have double. This one is single. So in the double data extraction, double screening, excuse me, you aim to have exactly the same setup on screening. What happens is that there is an algorithm, excuse me, that is working in the background. It will randomly and automatically allocate a paper to me as a user. It will ensure that this is available to another user for double-blind screening. And once you have double-blind screening ongoing for that reference across two users, you are not able to then have this pop up for anybody else. So the system is in place so that it allows you to have multiple people screening at exactly the same time. You could throw, 8 people on this project if you wanted to. The system will allow you to do that. It will automatically just pick out papers, and will then kind of lock it as needed um, when you complete that reference screen. So that's a really quick summary of ActiveSLR. I appreciate there's a lot more to it, and if you have any questions, that's very much why we're here. We'd love to have those questions, and there have been some that come through. So I'll hand back over to Shibata to lead those. And thanks so much for your time.
Thank you, Jan. That was really very insightful and helpful. There are quite a few questions came up, and the very interesting question actually. I think these are the questions that we have been also facing at ISPOR from the people you gave demo to. So I'll probably start with the very first one I see, and that came in just now. Is the PDF extraction accurate? I think that has been one of the questions we've faced quite a many times at ISPOR, if you recall.
Yeah, indeed. So it very much is. Now, it's not going to. We are not digitizing graphs, for example, so you wouldn't be able to do that. But the highlighting for your PDF highlighting, I believe that's what we're referring to. The PDF auto highlighting is indeed accurate. Those are identifying exact matches for the phrases, words, or letters that you have searched for. So one of the examples in that example I just gave was in psoriasis, where adalimumab is sometimes limited to ADA. Now, if you look for ADA, it will bring up Canada to have another ISPOR reference. It will do that. It will take it quite literally. Now, there is a lot of research going on into more advanced AI assisted data extraction of PDFs. Those are things that we're obviously monitoring very, very closely. What anybody who was at ISPO and maybe in many other conferences will also see is that while there are some people that are attesting to the accuracy of this, real world studies where people are truly trying to do full text data extraction from PDFs auto generated, they are not finding this is replicable, this is safe, this is something that you can do with any consistency. So we are very much of the opinion that at the minute this should be AI assist rather than AI replace. So I hope that answers the question. And yeah, so if it is, certainly one final thing to add is obviously more to follow. We're all going to be watching this field very, very carefully, and at some point in the not too distant future, I'm sure that everyone will be able to data extract from PDFs far more completely. But until then, it's AI assist from us.
Thank you. That's very helpful. I just wanted to add one bit there that while the PDF extraction that we see, the automatic summary, the AI produces a summary of its own. But what AI also does very helpfully is it identifies the line where you can find the relevant text. So the user is able to validate the text that AI is producing with the corresponding lines from the actual PDF text. So that's very helpful. Yeah, another question came through while you were responding to the first one. So the question is, does reconciliation address only inclusion-exclusion conflicts between reviewers, or does it also include disagreements over exclusion reasons in both primary and secondary screening?
Very good question. So Hale, I'll double check with you on this one, but I believe that that it is only on include exclusion decisions at the minute. It's something though that I think we're looking at. But Sale, please correct me if I'm wrong.
No, it is actually for all the study decisions wherever there is a conflict between both the reviewers, all those studies will be eligible for a reconciliation. So it could be to exclude decisions, both are different by both reviewers, those studies will come for reconciliation. No matter whether it is include versus exclude or exclude one versus exclude two.
That's right, excuse me. And we also looking at the opportunity to change that in a future version where you will be able to toggle between if it's excluded and both people agree, then fine. That's something that we're looking at. I got it the other way around. Thanks, Sahil.
Excellent. Now there is we received a compliment and a question all into one sentence. One of the attendees says, "Very good AI implementation. Other tools which give AI are so costly. How is this free?" Maybe I'll respond to that question. So the reason we are able to give this AI implementation for free, because what we have been able to do, we have been able to create GPU servers within our office premise, which allows us to provide some sort of AI capabilities on this app. So once this once somebody starts a project, then all the citations abstract gets uploaded to the system, and we have our internal AI server, which then looks into the text and then produces the AI Pico and so on. So because it is all internal and we are not relying on any external resources, that's why we are able to provide this for free at this point. And we hope that we would be able to continue doing this for near future and possibly forever. Okay. I will go to the next question then. The next question is, can I add multiple keywords in the full text?
Yes, indeed. Yep. So you can add in as many keywords as you want. Now, those keywords can be key phrases. So you could have relative efficacy as a single keyword, but also the number of keywords you can have as many as you wish. We haven't found the limit yet. So very good luck trying to find it.
Thank you. There's a question that we received from one of the attendees, which says, does ActiveSLR help in building search string or provide suggestions for search string? So at this point, ActiveSLR doesn't do that. ActiveSLR starts after you have completed a search and you have found out citations. But this is a good suggestion. We take note of it, and we'll try to see if there is a way to implement this where we possibly provide some recommendations or suggestions on the search string. But that's a very good question. We take note of that. Next one is from Praveen. And the question is, do you have more webinars planned after this one? If so, when we can expect them? Yeah, and I think you are the best person to answer this question. Over to you.
Yeah, thanks so much for the question. Great to see you as well. So we're certainly planning to do a couple of different things more. So firstly, I think we should very much plan for another webinar. We'll definitely look to see what topics people want, and so please reach out to us if you've got queries. I think that'll be really helpful for us to understand where the queries are, where we can help more, where the next webinar can help. I mean, it could well be something about data extraction, but equally it could be about some new features if we want to launch them, or it could be a more detailed focus on really the technical elements of ActiveSLR. So we would love to do some more. If people are up for it, we would be more than happy to do so. Please have this as a two-way dialogue, and we want to get people's feedback to be guided by how to develop further, and also how to develop those subsequent webinars. So please do reach out. The other minor thing I was going to say is, yeah, we'll also be putting together a series of smaller tutorial videos that we'll make sure we upload to YouTube. So there'll be a little bit of help, but very much not the end of it. So yeah, I look forward to seeing everybody here soon in the near future, hopefully.
Excellent. Now, I think the next comment is a mix of question and a little bit of complaint. I think attendees trying to say that there are other paid tools where even after deduplications, the studies still remain, duplicate studies remain, and the attendee has to remove them in the Excel manually, and which unfortunately affected the workflow in the past. So how does the deduplication work? I mean, what is the magic behind such an excellent deduplication?
Yeah, so I mean, I'll hand over to Sahil and Anoop for the technical element, but if I may butcher it momentarily, this is where kind of the AI assist comes into play. It's a large language model that is not just doing the direct checking that you see in something like EndNote, which is a very simple character by character check of duplication. There's also an element of understanding the percentage level of comparability, and therefore you have this determination of what is expected to be a true duplicate, but also there's this possible duplicate spell. So the aim is to have that. That's what the LLM does. It accounts for. I think the example I gave had like some errant capitalization or some differences there, or some differences in the output where it had like an Ovid in a notation which was erroneous in one of the duplicates. It's I the LLM is able to identify that as some type of syntax error and discount that, thereby ensuring that duplicate is indeed brought in. But yeah, so Hella and Noop, is there anything else there on the technical side of things as to um, how this deduplication process works?
I think, yeah, you summed it up very well. So I think one of the recommendation that we put forward for our users is that you can always go and by few clicks remove all the true duplicates, and then probably for the possible duplicates take a little moment to just read and check between the studies wherever those highlights are coming, and based on that you can give your decision whether to keep them or whether to remove them. But in totality, it's a foolproof method to remove the duplicates.
Thank you.
Okay. Uh, Meanwhile, we received other questions too. So Ian, one of the question is, can we add more exclusion criteria or it is fixed?
Oh no, those are fully customizable. Those ones that were there are presets, but those can be deleted. No, that is all fully changeable. I mean, one thing as well that we're looking into is the possibility of having subcategories for includes. The only reason that it's not in there at the minute is because we want to make sure that we don't risk end up steering towards clutter. You want to make this a very clean and crisp user experience. That's what we're aiming for, and we want to make sure that we don't lose that advantage that you get, hopefully, of being able to screen faster. That's where we know people save time. So but to the original point, no, all exclusion criteria are fully customizable.
Fantastic. And then there is another question which received two upvote from attendees. There's a question on, is there a plan to include .csv file?
I just noted that was a good idea. Anoop, Sahil, over to you guys. Well, I thought that was a good idea.
Yeah. Anoop, your thoughts on this?
Yes, so we are planning to have that in the next version of ActiveSLR, because in CSV we have multiple headers, and then we have to also ask the user to correlate those headers with the uh, places where we have those title and abstract and all of the other fields. So as of now, we only support NPIP and RIS files, but in future we will also support Excel files as well.
That's excellent. Thank you so much. Jan, while I was looking at this, and thank you for that excellent presentation, something that popped into my mind is how would you throw some light on what will be how attendees could manage their internal research projects by creating new roles and new responsibilities within the app? Or is it that completely fixed? Or will you
Yeah, no,
allow some
so
flexibility for them?
yeah, so those are all fully flexible. Now even the 3 presets that we offer to begin with, those are just based on what we use within PharmaQuant, but we know that everybody has different preferences. So no, those are fully flexible, and I think the example I gave was that you would be able to just to create a new role and change the permissions that you wanted for that role to have. That will immediately fill through to your entire dashboard, so you can immediately begin using that new role. That's something that you can also do and change as you go out throughout the project. So if you wanted somebody to move from a screener to a project manager, you can do that mid-project. Vice versa applies as well. And if you wanted to add in a new role as you go on, you can also do that. So there should indeed be full flexibility, and it should very seamlessly filter through as to you as a user.
Sure, thank you. Yeah, and another question came in while we were discussing. Is there any way the data extraction could be done by AI? And I think there is a lot of interest in what AI can do and cannot do. So over to you.
Yeah, no, it's the question on everybody's lips. The moment that AI can do data extraction is the moment that SLRs or any form of literature review can be done in extremely high speeds. Now, there are a couple of different challenges that come with AI data extraction. And by the way, this is definitely something that different people claim different levels of accuracy, superiority, and real world replicability. But the challenges that you see, and we saw this like over in ISPO, there were a couple of different posters that were showing real world uses of this, where they were taking RCTs that came from health technology assessment submissions. So some pivotal phase 3 RCTs. They were trying to use automated, AI data extraction. That was professing a very high accuracy rate of like over 90%, and it was just not working. Maybe some data extraction automation is possible in highly qualitative publications, but anything that we look at that has tables, that has figures, that has complex levels of science that require interpretation is currently not something that we have seen any form of AI extraction do at an appropriate level of accuracy and replicability. So that answer will be different for every single person that you ask, but it will be something that will also change over the next months and years. I'm sure we'll be sat here in a couple of years and the answer may be very different.
Yeah, absolutely. And to let the attendees know that it's not that we are not making an effort internally for this, and the research is currently ongoing within PharmaQuant, and we made some headway into this, but I think Ian said it right that the accuracy is always a challenge, and then replicability across different real world example is always a challenge. So I'm sure we'll have a breakthrough at some point. And so I want to second what Ian said there, that maybe in a couple of years we'll be sitting here and with a very different answer to that question. Ian, another question came in: can it manage high number of studies and high number of reviewer screening? Anup, you might be the right person given that you know very well what the system capabilities that we currently have. So how large the project can be, how many studies, and what number of reviewers can it support?
As of now, we can support a huge number of reviewers. So if you want to put me a number, it can be from 1 million plus reviewers on a single project. So, and you can have as much as of studies as possible imported if you are in a corporate or scholar account. There's no restrictions there.
Okay, so let me put some very outrageous numbers there and please confirm if they're correct. So you are saying that if there are 1,000 reviewers reviewing 1 million studies, that is possible. System-wise, it is possible. Probably there will be a project like that, but it is possible.
Yes, absolutely.
Okay. Another question came in, and this is for Ian. Do I need to wait till the screening is done to move to next step?
No, Nish, no. Everything is looked at by reference basis. So hypothetically, if you have screened and QC'd 10 papers, found the full texts, You could hypothetically, if you as a user wanted to, you could have data extraction, full text review, and title and abstract screening ongoing as you wish. Now, that flexibility doesn't mean that you have to do that. It just means that you can set it up how you want. You have the option to restrict as a team. You can say, guys, don't don't get full text reviewing yet, because I will the PM or I will only do my full text excuse me, my title and abstract QC once all the screening has been done. So you obviously need to have that QC step done, but as soon as that's done, then it's over to the next one at a reference by reference level.
Thank you. That's very helpful. I think that's all the questions that we have received so far. So overall, this webinar has been very informative and very interactive. Thank you all the attendees for asking those brilliant questions that made the discussion all the more meaningful. And thank you, Ian, for an excellent presentation. And again, thank you, Sohail and Anu, for doing a fantastic job and supporting Ian through this. If there are no more further questions, I think we can. Oh, there was another question came in just now while I was about to close. And I think the question is, can you show deduplication by running it?
Yeah, sure, I can do that. No problem. Right. Sure. All right, back to the examples. So webinar example. So this is the one here. So if I just run deduplication. Now, this will probably take, I would guess, somewhere around 60 seconds. It will depend on the complexity and the overlap that you've got in your list of papers. So if you have 7 uploaded text files, you have to do way more cross-checking. If you have a high number of references in each of those, it gets exponentially larger. So that's the point to make, that it has to do quite a high level of cross-checking. But what we're doing here is 1,000-ish duplicates being checked. If anyone's counting, let me know. I think this is a 2-minute job. Let's just see. The other point to note, by the way, is while this is ongoing, you can go and do other stuff. You can leave it on in the background. But what was that, a minute and a half, perhaps, I think, for just over 1,000. And that identified somewhere in the region of 300 duplicates. Hope that was helpful.
Yes, thank you, Jan. That was very helpful. I think we got a fair idea about what deduplication can do. Meanwhile, there's another question and there's a request actually. Can you share the recorded webinar link? Yes, this recorded webinar will be posted on YouTube, and then we are happy to share that YouTube link with everyone who attended the webinar today. So yes, that will be shared. Okay, thank you. I think we had a very productive discussion and very productive webinar. Thank you again, Jan. Thank you, Sohail and Anup, for supporting this. And thank you all attendees for making this discussion really interactive, and thank you for your patience and time. Hopefully,
Yes.
we will see you in the future again in the webinars. Until then,
Thanks so
take
much.
care. Bye-bye.
Cheers all.
Thank you, everyone.
Thank you, everyone.